Thank you everybody for joining. I'm going to start with a very short introduction for our journal club today. My name is Erin Crossey. I'm a pulmonary physician at Boston University, and I'm hosting our respiratory cell and molecular biology assembly journal club today. And I'm very excited to present Yujuan Su, who is coming from UC San Diego to present her very recent, earlier this month actually, Nature publication, which pertains to a subset of brainstem neurons which control allergen-induced airway hyperactivity. So I will let her get started.
Okay, thank you for the introduction. Thank you for the opportunity for the presentation today. Good afternoon, everyone. I'm Yujuan. I'm from Dr. Xin Sun's lab at UCSD. Currently, I'm a project scientist in the lab. For myself, I have a neuroscience training for my PhD study. So when I joined Xin's lab, which is a lung biology lab, a couple years ago for postdoc research, I saw this is a great opportunity to combine neuroscience with lung biology and trying to study the intersection between each. So that's why how we initiate and focus on the study that to understand the neural basis for lung to function as a sensory organ. I'm glad I have the opportunity to share with you our journey today and how it leads to the discovery about the neural circuit that is critical for allergen-induced airway hyperreactivity. So I want to show you with Lashi's movie. So this is about mouse lung. The green labels airway and the red labels sensory nerves. I hope you can appreciate that how widely innovation the lung is by the sensory nerve. So however, what's the function of those innovations and what's the neural basis for lung to function as a sensory organ is largely unknown, and this is the focus of my presentation today. So talking about a sensory system, I'm pretty sure everyone's familiar with the canonical five senses. We use it every day to sense the external world through the external sensory system. We can call it exteroception. So similarly, our body also have an internal sensory system, which helps our brain to understand what's happening inside of our body, including the heart, the lung, and the stomach. For example, I hope you are able to have lunch before joining the club, the journal club, so you have food in your stomach and you do not feel hungry. And talking about here, I can feel my heart beating is increasing. So all those stuff is called like interoception. So interoception defines a biological process about how the nervous system can sense, can integrate, process, and respond to what's happening inside the body, including the internal organs. And this internal sensation is in part carried out by peripheral sensory nerves that innervate the organs, including the lung, like the movie I showed you in the previous slides. So however, compared to the external sensory system, how diverse the interoception signal are coded at anatomic, molecular, and functional levels is really unknown. So this is the starting point that we address our question. So the lung stands out as an ideal organ to think about it as interoception organ because the lung has a bigger surface area. It is directly opposed to different kinds of environmental signals, including but not limiting to allergens, a lot of pollutants and smoke signals, and also like the flu virus, the COVID virus, and different level of oxygen, and also the airway pressure. And we know that the lung is innervated with a nerve, so this leads us to consider the lung as a sensory organ besides its canonical role in respiratory function. But how does it perceive the environment is unknown, and this is why we start. So in the next couple minutes, I hope I can show you our findings and how it leading to the conclusion, like it is a complete circuit that starting from the lung and back to the lung to control the lung function. And today I will use allergen as a model and study the airway hyperreactivity. So before our work, there are lots unknown about this circuit. For example, that what signals inside the lung can trigger the lung response, and how this signal is relayed through the vagal ganglia, and what happening in the upper brainstem, and how this information is finally transported back to the lung, to the efferent side to control lung function, and also what's happening inside the lung. So with all those unknown question marks, we started our project. To understand the interoception circuit, I think the first thing is trying to understand where are those neurons that innervate the lung. So we start with anatomical mapping. The strategy we use is we deliver the retrovirus, labeled here retrovirus, into the reporter, those are TB tomato mice. And in this case, all the lung cells is being affected by the retrovirus, including the fiber terminals. And then we trace back the neurons, and we found those neurons cell body, they are located in the vagal ganglia. Show them away. So if we clear the vagal ganglia, we can see that pretty a lot of neurons in the vagal ganglia are being light up, and then they can project a lung fiber that innervates the lung. So this means there is connection from the vagal ganglia to the lung. We also have previous study showing that in the other direction, that is, we deliver the reporter virus into different lines, which label subset of neuron in the vagal ganglia. And then we take the lung and we're trying to see their targeting or their projection pattern. And here is a brief summary. And we found there are different types of vagal neurons that innervate the lung, and they're targeting morphology, fiber pattern, and their targeting cells in the lung is different. So this is really fundamental work, but leads us to the functional study, like what are those nerves play the role. And remember, those vagal ganglia neurons, they are pseudo-unipolar neurons. That means one neuron, they can send bidirection fibers, and one targeting the lung, and one will go up to the brainstem. So what's happening in the brainstem? So the same strategy, we deliver retrovirus to the lung, and we label the cell bodies in the vagal ganglia. And this time, in the same mouse, we take the whole brainstem and we clear the tissue. This is left sheet image of the whole brainstem. We can see that the whole brainstem, only one region is lit up by the fiber terminals, which cause the NTS region. The full name is nucleus of the solitary tract. So that means lung innervating vagal neurons then project only to the NTS region for further information processing. So this is the focus of my presentation today about those NTS neurons. And the next question I want to ask is, okay, it projected there, whether it play a role, or in other words, can signal to the lung active those neurons in the NTS region. So I start with allergen, and I use a well-development allergen model called house dust mite. So this house dust mite, I easily challenge the mice. And after 4 weeks of challenge, I take out the brainstem. And this house dust mite, I want to mention, it can robustly induce the immunoinflammation and also the goblet cell multiplier, which indicating this is a robust allergen model. But to our surprise, in the brainstem, if we take a look at the NTS region, which is outlined in this shape here, so no allergen, which is sitting control, there is a very few cells express false. So false is a well-known marker for neuronal activation. Once a neuron is activated, it will express false. In contrast to the sitting baseline after allergen, we can see that there is a lot of neurons that express false, and the number is significantly increased compared to sitting. So that means those NTS neurons are being activated upon allergen challenge to the lung. And we also found that those NTS neuron activation is depending on chronic allergen challenge. That means it only happens after the fourth challenge. So after you challenge the mice with 4 doses of HDM in 4 weeks, you can see this activation is significantly increased, but not after previous doses, suggesting there is a plasticity or conditioning of this circuit. That means chronic challenge. So we want to confirm that this NTS activation is transverted through the vagal ganglia. The strategy we use is we're trying to ablate this vagal transmission through the vagotomy. That means you cut the vagal nerve on one side of the mice before you run the challenge. And you can see compared to some control, if you cut one side of the vagal nerve and in the same mouse, it takes the brainstem, you assay the NTS with false antibody staining, you can see the ipsilateral, that means the same side of the cut side, there is a significant decrease of false staining, meaning that this NTS activation means the vagal nerve transmit. And we also study about IL-4. So interleukin-4, IL-4 is known play a role in the allergen-induced airway response. So we're wondering whether IL-4 play a role in this NTS activation. So we administration anti-IL-4 antibody trying to neutralize those antibody, those IL-4 every time before we challenge the mice. And after 4 doses of HDM challenge, we see that anti-IL-4 will significantly blunted the false activation in the NTS region. So far, all the data suggest that NTS neuron can be activated upon the allergen challenge to the lung in a vagal nerve and IL-4 dependent manner. And then the next question we want to know whether this is important. So whether those active neurons in the NTS region play a role in any lung response to allergen. So we assay a lot of asthmatic responses, and we start with the airway hyperreactivity. So in rodents, airway hyperreactivity is classically being assayed by the flaccid assay. As you can see here, after intratracheal delivery of increases doses of methylcholine, while it can induce both increased airway constriction in both saline challenged mice and HDM challenged mice, it induces significantly higher resistance parameter compared to the saline in HDM group compared to the saline group, indicating the airway hyperreactivity. And this is mimic how acute triggers chronic heightened airway expedition in asthma patients. So we use those difference between the saline and the HDM group to as a readout the how hyperreactivity those airway smooth mast cells are. And then our question is, we want to know whether those NTS active neurons play a role in this airway hyperactivity. So the strategy we use is we're trying to ablate those active neurons in the NTS region before running the flaccid assay. So this is the strategy. We cross the first reporter-driven query with ROSA TD tomato as well as a ROSA different toxin receptor. So in this case, those active neurons will be labeled with reporter TD tomato as well as DTR receptor. And then we deliver the DTX, which is agonist, to ablate DTR expressing cells. We deliver DTX only to the NTS region specifically in those mice. So in this case, only the NTS region and only those active neurons will be ablated. And in those ablated neurons, we run the flaccid assay. And this is the data. As you can see here, those blue lines indicating the DTS ablation mice. And those mice, compared with their vehicle-injected control mice, they have blunted airway hyperactivity, indicating that those NTS neurons play a role in this airway hyperactivity. So before our study, there is a lot of studies showing that vagal neurons and also specific cell type of vagal neurons play a role in this airway hyperactivity. And our study is trying to go in and see not only vagal, but this vagal projection to the NTS region also play a role in this airway hyperactivity. And then the same strategy, we also assay other asthmatic responses, including immunocell infiltration and goblet cell multiplexer. But those two other effects do not affected by the ablation of NTS neurons, suggesting there may be other circuits participating in those two aspects, or there are just a local response within the lung that do not need the NTS participation. But those all need further studies to validate. And following this, we want to ask, okay, the NTS neuron play a role, but what is the molecular signature of NTS neurons? Because NTS neurons is really heterogeneous and it receives input from almost all the internal organs. So we want to know which cell type of the NTS neuron play a role in this allergen response. So we set out to use a single nuclear RNA-seq technique, which can provide NTS cell signature at single cell level. So this is a workflow how we did this. We grab the NTS region by fresh dissection, and we're trying to avoid nearby, close by nucleus, trying to avoid the contamination. And after this nucleus isolation, we do the 10x RNA sequencing. And in silico, when we analysis, because there are available other data sets, for example, the nearby DMV and AT regions, so we can purify the data and trying to make the data is really NTS specific. And in total, we have 32K NTS neurons from 6 biological replicates. And we are pretty confident about our analysis. And this UMAP showing here, there are in total of 18 clusters in our NTS data sets, including some excitatory and some inhibitory neurons. And we are also able to plot a distinct marker genes for each of this cluster as listed here by dot plot. This gene list is really useful because in this case, our question is we want to know which kind of those types express false after allergen challenge. So we can use those marker genes together with false and run in scope in situ hybridization, trying to match which is the highest overlap among those different types of cells. And this is the summary data. So we're trying to do false RNA scope together with each of these 18 cluster marker genes. And we found that cluster 13 overlap the highest as labeled here. So cluster 13, the marker gene, top marker gene is DBH. The full name is dopamine beta hydrolase. As shown here, we can see that there are a lot of overlap between false and DBH. So that our focus zoom into the DBH gene. And we want to understand why the DBH neuron really play a role. We have different kinds of approaches, including toxin ablation using the chemical toxin. We also have genetic ablation using DBH-Cre crossed stereotopsin receptor. And we also have the chemogenetics. So all those approaches have the similar same results. So today I'm just showing you the chemogenetic approaches that we used. We call it a DRAG system. So DRAG system is that you overexpress the modified muscarinic receptor into the regions of your interest. And there are two forms of this receptor. One we call M3D, one we call M4D. So difference is M3D, once it is expressed, it can activate those neurons. And M4D, on the other hand, it can inhibit those neuron activity. And all those activity increase or decrease is actually controlled by a drug delivered called CNO. So in this case, we can decide which cell types we want to modulate and when we want to control those activity. So with this strategy, we start the experiment. We start with the M4D inhibition version. So M4D can be delivered through the virus version. So this is Cre-dependent M4D. We inject this to the NTS region of DBH-Cre mice. So in this case, only the DBH neuron, only those DBH neurons in the NTS region will express the inhibition M4D protein. And then we deliver the mask with CNO, trying to inhibit those neurons during the allergen challenge. And this is the functional readout, the flexibility data you can see with the CNO, which means those neurons are being inhibited, and you can significantly decrease the airway hyperreactivity. That means those activation of NTS DBH neuron is necessary for allergen-induced airway hyperreactivity. So once those neuron activity is off, the airway hyperactivity is decreased. On the other hand, we want to know whether we activate those neurons, it play any role. So in this time, we deliver the Airway and create-dependent M3D version of the protein, which is activation version of the protein, into the NTS region of DBH cre mice. And after CNO deliver, in the absence of HDM, only CNO deliver. That means only activation of those neurons. You can see in the middle line, the yellow line, we are able to sufficiently increase the airway hyperreactivity above the line of the seeding control. So that means this activation is partially sufficient for allergen-induced airway hyperreactivity. So once those DBH neuron activity is on, you can increase the airway hyperreactivity. And for all those modulation of NTS DBH neurons, we also assay the breathing rate and heart rate, and we didn't see any significant effect on those other parameters. So that means those modulation is pretty specific to the airway hyperreactivity in the lung response. So then the next question is, we want to know how those DBH neurons play a role to modulate the lung response. What's the downstream pathway of those neurons? So we use the virus treatment strategy to address this question. The way we did is we deliver the query-dependent reportovirus, to the DBH query NTS. So in this case, the DBH partner neuron in the NTS region then will express FOS, and their fibers will targeting to other downstream nucleus. And in the same mice, we take the whole brainstem, we found that the NA region, which is lit up by VHH, as you can see here, it can be innervated by those fiber terminals. And those fiber terminals are projected from the NTS regions. That means the NTS region or the DBH partner neuron in the NTS region, they can directly targeting the NA neurons. So whether NA neurons play the role, whether NA neuron can transmit the information back to the lung. So this time we use the same strategy, but we're targeting the NA neurons. Also NA neurons then express chat. So we use chat query and try to capture all the NA neurons with the Airway query-dependent reporter. And in this case, we take the airway. So this is the upper airway. We take the 6 section, we see that there are dense fiber innervation of those parasympathetic neurons lit up by the chat here. And those neurons receive intense fibers from the NA regions. So that means if you trace NA neurons, you can see fiber terminals in the upper airway. So structurally, they are connected here, but whether functionally they play a role, this is our question. So to understand whether the NA neurons, they also play the same role in regulating the airway hyperactivity, we also use the DRIDE system trying to inhibit or activate those NA neurons. So this is our data you can see here. If you inhibit those NA neurons with the CNO, and you can significantly balance the airway hyperactivity by the HDM. However, if you activate those neurons in the NA regions, in the absence of HDM, only the CNO, again, as you can see the middle line, you are able to partially sufficient to increase the airway hyperactivity. So that means those NA neurons is also important. They are also necessary and partially sufficient to mediate allergen-induced airway hyperactivity. So the check mark here. And then the last question and the most important question is we want to ask, like, what is the transmitter in between? How the NTS neurons and NA neurons being connected. So what is the molecular signature? Because we have the NTS sequencing data, we also generate the NA sequencing data. So right now we can in silico to build a ligand receptor pair from the NTS to the NA. And in this way, because DBH is dopamine beta hydrase, this can convert dopamine to non-epinephrine. So we also look at non-epinephrine receptor in those NA regions. And in this case, we found non-epinephrine may be a possible or potential candidates that can commute the information from NTS to the NA. So we focus on non-epinephrine, and we use two specific non-epinephrine receptor antagonists, those two shown here. So we give the match HDM, but in the same time, we're trying to deliver the non-epinephrine receptor antagonist in the NA regions. And we are excited to say that if we block the non-epinephrine receptor in the NA, we can actually blunted those airway hyperreactivity, meaning the non-epinephrine is really the transmitter. And we further confirm non-epinephrine is the downstream of this DBH positive NTS neuron. So this experiment is we're trying to activate those DBH positive NTS neuron through the dry and M3D strategy. And in the same mice, we deliver the non-epinephrine receptor antagonist in the NA regions. So that means we activate the upstream, but we block the downstream neurotransmitter transmission. So if non-epinephrine is really the K, in this case, we will block the transmission and we will block the airway hyperactivity. And this is the case we see. So this is the summary that DBH positive NTS neuron, they really act through the non-epinephrine to control the airway hyperactivity. So far, this kind of summary of the data I show you here. So we're trying to find a neural circuit, a full circuit that starting from the lung, once there is allergen challenge to the lung, how this information is transmitted through the vagal ganglia to the NTS in the brainstem, and then transmitted to the NA in the brainstem, and then back to the parasympathetic neurons in the upper airway, and those neurons control the constriction of the airway smooth muscle cell. And in each node of this circuit, we're trying to give the molecular signature and trying to give a more detailed understanding of this circuit and how this information is transmitted. And we hope with this molecular signature and those non-invasive neuromodulation maybe as a potential way trying to more specifically or more efficient to control the asthma treatment. So this is the direction that we are trying to work towards. So far, this is the data showing our current work. And I'm also running some ongoing work, which I like to share some of with you today. So the NTS region, so as this shown here, the NTS region is being activated by allergen input to the lung. But NTS region not only can project to the NA regions within the brainstem, it also can going up to higher brain. So PVN, which called paraventricular nucleus of the hypothalamus, is one of the downstream targets of NTS region in the higher brain. So I also see that there is a significantly increased force expression in the PVN region after allergen challenge, which means that PVN region is also being activated in asthma mice. And this PVN activation is very interesting. So it's depending on the NTS activation. As I mentioned to you, so this is a downstream target of NTS region. So if you ablate those active neurons in the NTS region, you will significantly ablate the PVN neuron activation. And the PVN neuron, this is a very famous region, which controls HPA axis, a stress hormone release location. So then I want to see whether those P1 activation play a role in allergen-induced stress response. So I use behavioral assay. I found that in our current HDM challenge model, the mice will behave some stress behavior. But if you ablate those active neurons in the P1 region, you can reverse those stress behavior. So that means this P1 region is actually modulates the stress response in asthma mice. So this is just a brief summary of the hypothesis that I'm working on. So I thought once there's allergen change to the lung, it not only activates the NTS region in the brainstem, which modulates airway hyperreactivity, but also this information is transmitted to upper brain in the P1 region for further information processing. And this activated P1 region play a role in the stress response. I'm running I'm running future work trying to figure out the molecular signature and how it modulates the stress. But I thought this is a great point trying to follow up. So in future, I think not on the allergen as I talk with you today, but there are lots of different interception signals in our environment, as listed here. So it will be interesting to study like how or whether there's any difference between different circuits in response to different interception signals. And I want to address those questions in different ways, trying to anatomic molecular mechanism, and then trying to capture their molecular signature, as well as trying to give them functional regulation. Okay, so this is my acknowledged slides. So I'm glad to have Xin as my mentor along the journey, and she is very supportive, and I'm glad that we make the way out. And I will thank all the members in the Xin Lab for their discussion and help. And I have a lot of collaborators inside the UCSD and outside of the university. And I also thank about my fellowship. Okay, so one last slide. I'm now in the job market. So if you know any job opening or if you want to follow up with collaboration, please reach out to me. I want to emphasize here like my research area, because I have a lung biology training in my postdoc and I have my neuroscience training in my PhD. So I thought it is a great opportunity to integrate those two aspects and trying to study the brain-body interaction. Not only asthma, as I mentioned here, but also other environmental signals. So thank you, and I'm happy to take questions.
All right, thank you so much. That was a great presentation, and I am happy to kind of open it up to questions in the chat.
Yeah.
I myself and also Alan Fine and Dan Aridgides are here as moderators, so perhaps we can sort of start with a few questions while people may also add them to the chat. And I guess I can start with one sort of question out of just sort of ignorance with neuroscience, which you obviously have a background in. Is there sort of pathologic data in humans, for example, in like brain tissue banks, that you are able to sort of corroborate your mouse data with, or is that something that doesn't exist yet?
Yeah, this is a great question. So when I start the NTS region, I want to start with whether there is any human data, for example, MRI data, seeing whether there is activity increase in the NTS region in asthma patients. I know there are some cough patients near NTS neuron, and all the downstream nucleus of NTS region, they are being activated by the MRI data. Allergen, I have to say that, but I know there are some people working on that. But it may be a little bit hard because the NTS region is very small, right? So you need a bigger resolution trying to zoom in and see this NTS region. And the mouse model there to give us a good opportunity trying to study the molecular cell type. But yeah, I thought it's a great question to connect to the clinical data.
Dan, I have a few more questions, but I can wait my next turn maybe, if Dan or Fallon have a question.
I have a question. Do you think that there may be other physiological effects, or are there other places that control physiological parameters like respiratory rate and vital capacity, et cetera, et cetera?
You mean the NTS region or the DBH?
Region, during illness, or whether that's also whether there are distinct regions in the brain that regulate those sorts of physiological parameters.
Yeah, I'm sure there are, like the prebundal complex. It is a very famous brain center, right? It contains the brain, and also the NA region, which is called nucleus and bulgus region. It not only projects to the lung, but it also projects to the heart. And there is paper from Mark Krasno's lab saying that those NA neurons also play a role in the heart beating, those things. And for the NTS region, because I know that NTS region, there is previous research saying that FOXP neuron in the NTS region, it can control breathing in mice. So when I run in my those modulations, I also pay attention to those respiratory effects or even heart rate modulation. But those DBH neurons seems pretty specific, or at least to my modulation. So it only play a role in this AHR modulation. But I'm sure there are different circuits, like or even starting from different cell types in the same nucleus, maybe play a role in the respiratory modulation.
Thanks. I have a question, if that's okay.
Yeah.
Very nice work. I had actually kind of several related questions about the involvement of the vagus nerve in these responses. Obviously the vagus nerve innervates almost all the visceral organs, if not all of them. I don't recall exactly, but how do you see the specificity working within the lung? For example, would activation of the vagus nerve by intestinal stimuli cause a similar response? And also, how do you separate out the efferent effects of like the vagus nerve activation of muscarinic receptors in the lung, which can also cause, mucus production and asthma-like phenotypes?
Yeah, this is a great question. So I think this contains two parts of questions. So one part is how specific those different types of vagal neuron control different organs and different response. So in our model, we also have following work trying to figure out the cell types in the vagal neuron that specifically go to the lung play a role in our circuit. So we think the AAA is the type that's in our circuit. This not only by our work, but also by some other previous investigators like Dr. Ruka Charles and also Dr. Wolf's data. They also say that AAA1 may be the population. But as you said, AAA1 vagal neuron not only go to the lung, but also to other organs as well. So how this information of lung specific going up to the NTS region, I think there may be some receptors that specifically ligands released from the lung cells or even immunocells within the lung, and receptors being activated by the vagal neuron and then initiate the circuit. The other question is about the vagal sensory nerve and the efferent nerve part. So yeah, I know that there is a lot of data showing that vagal nerve stimulation mostly play a role through the efferent nerve going to the modulate the lung. So for us, I thought this is a nerve that's projection from the brainstem, the NA region. passing through the vagal bundle and targeting the parasympathetic neuron in the upper airway that play a role here. All those neurons in the upper airway, as well as those neurons in the inner region, they are checked. So no doubt they will release acetylcholine, methylcholine, and then they will activate the methylcholine receptor smooth muscle cell in the lung. So I think this is how specific, like the sensory nerves sends the signal to the lung and then process in the brainstem and functional through the vagal efferent nerve to release methylcholine to regulate the smooth muscle cell in the lung. Yeah, so how specific or whether there are different kinds of the efferent nerves, for example, whether there are different kinds of cell types within all those check positive neurons in the upper area, I think my answer or my guess is there are multiple types. So which one is the specific one? I think it may need further study. Yeah, this is complicated, but you have to work to focus on the specificity in each step. Thanks.
I guess a little bit, even just because it's on this slide here, but so do you have any plans to kind of investigate these other sort of stimulants? Like, I guess, for example, like cigarette smoking seems like a nice, very ready sort of clinical application.
Yeah, this is exactly what I'm trying to run. We have a collaborator, Dr. Lara, in here, she is very good at the e-cigarette exposure. So her lab has the setup. So I think my starting point in trying to expose the mic to e-cigarette exposure and trying to see what happening in the brainstem or the brain. But there are a lot of study from human as well, seeing that e-cigarette smoke exposure can affect the brain response and also the immune response. So there are studies there, but I guess the circuit or like which is the first region in the brainstem that light up by the exposure and then initiates the following response. I think this is unknown, and this is pretty important if we want trying to block this information progression. So yeah, this is something that I'm thinking, trying to work with her.
And I guess this is kind of maybe off topic and not involved in like any of your prior work, but just do you have any knowledge, like what's the timing of development of this whole circuit, you know, from airway to the brain and back again in terms of like just stages of development?
You mean the timeline?
Yeah, like at what point in normal development is this sort of fully intact, if that makes sense?
Yeah. I think so I think the question is whether this circuit only exists in adults or whether it is like development. It is very interesting, like the vagal neuron, you know, it's fully developed, I would say, based on the size and all those neuron projection, super early. I would say like neonatal, there are vagal neuron fully developed in the nerves, this innervation in the lung pretty beautifully already. And neonatal, the brainstem is actually fully developed, the size, and there are recently some paper using the confocal image, the calcium activities in the NTS region. And in their study, they are using like P14, even very young mice. And they already see that the NTS neuron is firing up in response to different visceral inputs. So I think the circuit maybe exists super early or earlier than we thought. But as we know, like the brain is still undergoing like development, right, till the adult. So the higher brain, like the PVN part, may still need the information processing part may still need a long time to be mature. But I thought those from the vagal to the NTS and then back to the lung, it may be like earlier.
All right, I think there's a chat question, which I'm happy to read, from Mats Johansson. So he said, if you didn't address this already, can you say something about airway hyperresponsiveness in mice and humans? People seem to believe that they're completely different things, and that data on AHR in mice don't say anything about AHR and asthma in human patients. Do you agree or not?
I would say, like, I'm sorry, I know little about what AHR like measurement or what is the parameter in human patients. But I'm sure the assay we use now is pretty well known or well used in the rodent study. So yeah, I don't know whether there is any difference between the AHR measurements in mice and human. So I cannot say agree or not because I have no information about this measurement in human.
Yeah, that's a good question. I don't know if any of my co-moderators have any other insights. I think that the metrics, the readouts you have shown were the ones that I would be familiar with most.
Yeah.
Most sort of
Yeah, maybe in human there are more like parameter you have to reach besides the constriction part. Yeah, but I'm happy to learn if you can yeah.
I mean, there's obviously the methylcholine challenge testing in humans, which is somewhat similar. We just use FEV1 drop instead of the airway oscillometry that I think is typically used in the mice. I'm not familiar with like asthma mouse models and sort of what are the challenges in sort of applying those to humans, but
and I think Dan or Alan, do you have any other questions, or anybody in the audience as well? I'm happy to read questions from the chat.
I had one more question about the prazosin and terazosin. How were those delivered orally to the mice, and like do those cross the blood-brain barrier? Because I did a quick PubMed search, and it looked like there were a couple of papers from like 1980 on using prazosin to try to treat asthma, but you know, given that that's a semi-well used medicine, it seems like if that was really effective in treating asthma in people, that we would probably have seen something by now.
Yeah. So the way I deliver it, I plant a tube into the brainstem region, into the NA directly. So yeah, this is the thing I'm also worried about, this BBB permission ratio. So I just deliver locally to the NA region to get this maximum effect. Yeah.
So if there are no more questions, I think we very much appreciated hearing from you in this exciting new publication. I think, did you have your email and information on a slide? You mentioned you were in the job market. You can
yeah, I should put my email there, right? Yeah.
But that would be something we'd be happy to highlight today for you.
Okay.
Yeah, thank you so much. You're also, I also have that information, so we can.
Oh, thank you. Yeah, yeah. I forgot this important email information, but yeah, thank you. Yeah.
All right. Well, I think if there's nothing else, that will conclude today's journal club. Thank you for everyone who attended. Recording will be sent to everyone who registered and also is going to be available on the ATS website at the RCMB assembly. I think journal club portion. So thank you so much, Yujuan.
Thank you. Bye, everyone.